Fortunately, I was very rude to our speaker by keeping the first one long, but the speaker doesn't mind. So thanks very much for actually asking me to speak and talk about some of the work that's been going on in my lab the last few years. This is a new talk that I've put together that hopefully you'll see the clinical relevance of some of the basic science work we've been doing in the laboratory. And the question that we are interested in lately, we saw this throughout the Grand Rounds presentation today, was how so many things, skin manifestations, skin disease, seem to be associated with systemic illnesses. So I'm going to give you a little brief overview of that topic. These are my disclosures. None of them are relevant to what I'm going to be presenting today. So most of you have heard the name Hippocrates, and those of you with MDs have taken the Hippocratic Oath. You may not know, actually, that Hippocrates was probably his main medical activities were as a dermatologist. You know, that was at the time, one of the principal methods in medicine for diagnosing patients. And some of the terms that we use differently today, when used by the ancient Greeks, really related to derm specifically. So the term idiopathic, its Greek translation anyway, meant originating from the skin, not its current use of originating from doctors. And then rashes, when you talked about a rash, that just meant to the ancient Greeks that it originated from a systemic disease. In the modern era of dermatology, these types of associations became more refined. And even about 100 years ago, and the relative underrepresentation of women in medicine, there was a really brilliant dermatologist. She was started in Germany. She was Jewish. She fled during World War II. Some of you may have heard the name. If you haven't, hopefully you'll remember this, but that's Helene Kurth. And the Kurth criteria became commonly used to describe skin manifestations that could trigger an interest in looking deeper for cutaneous malignancies. And she's the person who came up with the idea of malignant associations with the skin manifestations like acanthosis nigricans and the sign of Lesser Trellat. Now, since then, you know, there's some controversy, particularly in Lesser Trellat, which is multiple seborrheic keratoses, whether those are a very accurate assessment. But she certainly made major advances to start getting dermatologists to start being on the lookout that maybe they can make a systemic diagnosis for something that seems like a trivial cutaneous presentation. So I'd like to just start, just in general, asking folks in the audience, what, particularly the new residents, what types of skin diseases do you know that are associated with systemic diseases? And don't bring up any of the ones we talked about yet. Anyone can volunteer. Don't want to pick on anybody. Someone, yes. Okay, calcium, yeah, so we've got this deposition of calcium in the kidney. Well, that's really interesting. So prurigo nodularis is an itch, right? But it's a scratch, and you think that's associated with any sort of brain? Yeah, right. So we have beliefs about what the pathophysiology might be, and when we talk about skin diseases, we kind of make up in dermatology something to explain it, but that's great. Other stuff. Sarcoidosis and? Okay, so that's the same disease manifesting both places. Yeah. Erythema nodosum and IBD. Great, thank you for bringing that up. Inflammatory bowel disease and E. nodosum. Tricky connection there.
Bye, Judy.
All
Have
right,
a
so that's a kind of a beginning. There's many more, and I think you know them, but you're being shy, but that's okay. Um, So clearly the consensus opinion in dermatologists and hopefully in other specialties is that observing the skin can be a useful method for diagnosing systemic diseases. But really what we've been thinking is that understanding this in greater depth really would permit us to understand more about the pathophysiology of both the systemic disease and the skin disease that could lead us to understanding better ways to treat it. Because we tempt to, we wave our arms saying, oh, let's say we get a inflammatory panniculitis because of a pancreatitis, and we explain it by these systemic pancreatic enzymes that could do things, but why is it in specific locations? Why does it develop these nodules? And there's a lot of unanswered questions in these kinds of associations that we'd like to try to help answer. So in this presentation, I'm going to go through three subject topics led by very talented people in my laboratory. They're looking at specific aspects of the communication between skin and systemic disease. The first is stress and skin infections. So that's a little bit in terms of what you're talking about prurigo nodularis, but there are many other things that stress can trigger in the skin, of course. Inflammatory bowel disease and skin inflammation, and then psoriasis and cardiovascular disease. And we'll talk about that a little bit more when we get to that section. And our approach, the way many scientists work, is that there's lots of things you can think about, but we're biased. And our approach was to be biased on the types of topics that we've been studying in the laboratory. So in order to explain our findings, I'm going to spend a few slides talking about a little bit of a history of the work we've been doing in the laboratory and why we focused on this specific way to answer part of the questions of these associations between skin and systemic disease. And what my laboratory works on is innate immunity. Innate immunity has become defined as that aspect of our host defense system that's genetically programmed. There's no need to learn. Your innate immune system can change over time, but it's not necessarily being educated. And some of the notable people who made major contributions in this field won the Nobel Prize for it in 2011. Bruce Boitlow, who was here on the Mesa, was actually a collaborator of us, who through a genomic approach, an unbiased genomic approach in mice, found toll-like receptor 4 and sort of broke open the innate immune receptor recognition field for mammals. Jules Hoffman is also a friend and colleague at the Pasteur Institute in France. He did a similar thing, but in Drosophila, where the genetics are much faster, and he found many of the signaling systems that could later be translated in man. And Ralph Steinman was an adaptive immunology dendritic cell person, so he doesn't count. From this work, at the time when I was really just actually in my postdoc, we started an unbiased approach in skin wound repair, looking for molecules that could be involved in innate responses, and got lucky to discover that these things called antimicrobial peptides exist in the skin. And we've since learned that these types of gene products are expressed everywhere. In mammals, they're expressed almost in every tissue, certainly heavily used in epithelial tissue. And we continue to learn things about how our cells are programmed to produce natural antibiotics that help resist infection. And even one of the cell types that do that is the fat cell, as it differentiates from the fibroblasts to mature fat. These antimicrobial peptides, or AMPs, are very, very diverse. They have many different structures, but just as a primer, as a simplified way to think about them, is they don't work like a growth factor or a cytokine where it binds to a receptor. Instead, they have special properties that allow their interaction with the membrane to change the membrane. And when they're interacting with a fungus or a virus or a bacterium, that interaction can kill the organism or at least stop it from growing. But as you'll hear later, that interaction can also do other things with other membranes. So it's a broad, very, very complicated membrane active interaction that we're just starting to learn more about. One of the interests in my lab starting off early and as a dermatologist was to try to understand if these things that we actually originally found in pigs and then cloned the gene in mice are important to human disease. And with a lot of very great collaborators, we're able to show that in atopic dermatitis, the antimicrobials are suppressed by Th2 signaling, and that explains in part why they get colonized by high levels of Staph aureus and other bacteria. It helped break open understanding of the pathophysiology of rosacea, because one of these antimicrobial peptides will induce angiogenesis and flushing, and we found that patients with rosacea have too much of the gene expressed as well as the enzymes that process them. And then there's stories in psoriasis for how it induces interferon. This molecule can also promote photosensitivity. And then most recently, actually, one of our collaborators working with us up at UCLA using a machine learning approach for how the AMPs work that are naturally programmed, he discovered that the mutations in the COVID virus, in its coat protein, some of the proteins on there started resembling our own antimicrobials and causing the COVID virus itself, that coronavirus, to be much more inflammatory, trigger a much more robust response. So we think that's part of the reason why this cold virus became so deadly, because it mimicked the antimicrobial peptides. So the antimicrobial peptides, as I mentioned, are kind of everywhere. They're many, many different gene products, different structures. But one of the important things to recognize is that all forms of life produce the antimicrobial peptides, including microorganisms. And the microbiologists had known that, but in terms of skin relevance, Teru Nakasuchi, who's sitting in the audience, yay Teru, came up with the ideas, let's do a blind screen of those microbes that live on the surface of human skin and find out if there's any association with their ability to make antimicrobial peptides that could benefit us. And with very, very hard work, he discovered all of these new genes that are expressed by specific strains of some bacteria that can kill Staph aureus and kill other things too, depending on how you do the screen. But he found that one of the big cell types that are producing antimicrobials on our body are just those microbes that live on the surface of our skin. And we've taken that, and with the help of collaborations with Dr. Hada's group, into the clinic and shown that by putting those bacteria back on atopic dermatitis patients, you could reduce their Staph aureus and appear to start to improve their course of disease in atopic dermatitis. And right now, Dr. Hada and 8 other institutions around the country are doing a large-scale clinical trial funded by the NIH still, to test this in a long 14-week double-blind placebo-controlled trial. So we're looking forward to the result of that in the next couple of weeks. And what's interesting is you do this stuff and all of a sudden you start talking about probiotics. And now, although we've been doing the basic science, which we were really excited about, the news gets interested in this. And there was a lot of news published on this kind of probiotic work. I hate using the term probiotics because most of it's baloney, but we think in this case, actually, there's something really scientifically relevant there that we can exploit. Okay, so back to the topic. So that's the background of innate immunity, these small molecule communications, how they're affecting microbes, how microbes help us very, very quickly. How do we translate what we've learned in this innate immune field to asking these questions about how skin communicates with systemic disease? So the first vignette is the communication between skin and gut. Most of us, and certainly the lay population, think that you eat something and something happens in your skin and there's the wrong microbes. She didn't take the right bologna probiotics, and that makes your psoriasis get better, your hair grow, or all this other crazy stuff that they make billions of dollars selling. But the bias has always been the skin microbiome drives human health. The way that works in the skin is rather loosely defined. There have been some molecular mechanisms proposed to explain them. A lot of it has to do with the metabolites of the microbes that are in the gut. A lot of these are short-chain lipids that have very potent effects on gene expression and behavior of the immune system. Some of this may be true, but quite frankly, a lot of it is not well proven and very, very speculative. But a lot of work, a lot of investment has been made in trying to understand how the gut controls the skin. But we're kind of contrarians by nature, and we wanted to ask the opposite question. Is the gut the slave of the skin? Is the gut responding to skin things? And that question was really prompted by the entry into my lab of Tatsuya Dokoshi, who's now got his own lab and program over back in his home institution in Japan, who was not trained as a dermatologist, but trained as a gastroenterologist in addition to his PhD. So I said, oh, Tatsuya, you know about gut. I have this question. Why don't you test it? Why don't we just do a simple experiment? It's not too expensive. Let's take a mouse and make a cut on its skin. So we'll do something very localized, specific to the skin, and compare how a model of inflammatory bowel disease looks in this mouse if it has a skin cut, a skin wound, or no skin wound. So that model is something called dextran sodium sulfate. They just drink this high molecular weight solution, and it causes injury to the colon. So it's a type of colitis. And when he did that, we were shocked because when mice had skin-specific intervention, the colitis model killed them. They died. About 60% of them died. Whereas if they had normal skin, normal healthy skin, they lived. So it was very clear to us that there was indeed a communication between the skin and gut. And you could see, in addition to the life and death outcome, the histopathology was different in the colon, and the inflammatory cytokines were very different in the colon. So we love big phenotypes because it allows us to test a lot of different hypotheses. And you flash forward a few years of a lot of hypotheses that didn't work, okay? So a lot of failed experiments. But we eventually went back to some of the innate immune things we've been doing for several years that had found that when there's a skin wound or skin inflammation, your extracellular matrix digests itself somewhat. And hyaluronan, a large molecular weight glycan that we use as filler, gets chopped up by an enzyme. And those little fragments activate the receptor that Bruce won the Nobel Prize for, TLR4. So we found this sort of way to trigger systemic innate immunity by a skin-specific inflammation. And without showing you all the papers and all the tests, basically Tatsuya proved that's how the skin is making the gut different. It's because these fragments of hyaluronan circulate and the gut detects it, and it changes. But that's not enough to really explain why they should die, right? It's just, when you cut the skin, the colon appears normal. Mice don't get colitis. They just have a difference in their susceptibility to other hits. So we next asked, why would a danger signal from the skin change the susceptibility to colitis? And we use this model again, but now having learned that it's the hyaluronan, we had a genetic model in mice where we could just express an enzyme that cut the hyaluronan in the skin and release those fragments. And in this situation, there's no other complications like the mice are stressed from a cut or there's an inflammatory response because these mice are normal. They're just spewing hyaluronan fragments from the skin. And when we did that, now using, we flash forward about 4 or 5 years, we have better techniques for analyzing tissue responses. We use the single-cell RNA sequencing approach. You've heard about that from some other speakers in grand rounds over the years. Most of you probably know it, but basically what you do is you take the tissue, you digest the cells, and then you individually determine what genes are expressed in each cell separately. And computationally, that's a challenge to learn about all of that data, but doable. And many of my people in my lab now are really good at it. And when we did that and combined it with spatial sequencing, which is sort of the same approach, but now we look at the genes in the tissue itself, where they're located, Tatsuya found that 2 antimicrobial genes were changing in the gut when we did these skin-specific inflammatory responses. And those genes here, in one case, was showing this thing called Reg3B, which is an antimicrobial protein. There was another gene called MUC2, which is the mucin-producing gene. These are super important for the gut. But when you do any kind of skin inflammation, the gut's response is, oh, there's danger, danger, danger. Let's change the expression of these antimicrobials in case the danger gets to us. And we could validate that then after doing the unbiased genetic approach by just immunostaining. And what this swirl is the mouse intestine rolled up like a jelly roll. And the red is showing the expression of the Reg3 protein. So you see in the mouse that has the hyaluronidase, but nothing else. It's making tons more of the protein. The green is the MUC2. That's also different, but a more subtle difference from control. So the antimicrobials change in the gut after a skin effect. So if the antimicrobials are different, does that change the composition of the gut microbiome? Which I kind of implied that the gut probiotic stuff is all baloney, because in truth, the stuff they sell you is, but the reality of it is the gut microbiome is very important for systemic health. So we needed to know if there was a dysbiosis. So what is dysbiosis? What that means is that the composition of the microbes slightly changes. And these are complex communities with many hundreds or thousands of different species and strains of microbes doing different metabolic processes. You might not see actually a change in the total number of bacteria, but instead of only 1 red bacteria as illustrated here, you might get 3 in the new environment. And maybe a new one shows up, like the yellow one here, which I'm using to represent Staph aureus, which appears in gut dysbiotic communities. And that's very different than an infection, which would be defined by us as true disease, where typically what you have there is one specific strain just greatly expands, so you have a lot of Staph aureus. And not only that, penetrates through the epithelial barrier. So we saw no evidence of infection in our mice, but we wanted to know if there was a dysbiosis. And to test that, you do microbiome analysis. Again, I've talked about this in years past. You may have heard talks about this. But again, what you do is you sequence all the DNA. You understand what types of strains of the different bacteria are present. And from that sequencing, you can predict what types of metabolic products are produced. And what we found was that, not sure if you can read this, but some of the metabolic products that are associated with disease were overrepresented when there was a skin injury. And some of the metabolic products, like these short-chain fatty acids, which are associated with health, were decreased. So there was a clue to us that the skin injury caused the antimicrobials to change, and when you had an abnormal antimicrobial balance, then the microbes were changing, and now the microbes were doing things that were maybe not helping gut health. And when we looked at the gut, we could see that the microbes were actually now, the community there was, the ones that survived the change in antimicrobials were more dangerous. And the yellow dots deeper down there show that they're penetrating through the mucus layer, and that can be quantified there. So is that the answer to the question? Is that why skin causes the gut to become more susceptible to colitis? And the simple test was to actually do that fecal microbiome transplant that you've heard about. So we took mice with skin wounds and normal mice and collected their poo and then gave it to mice that had no skin wounds, identical mice. So the only difference between the two mice would be the microbiome itself. And when we did that, we confirmed the hypothesis, and that the mice that received a fecal microbiome transplant from a skin injury mouse, those mice got worse colitis than those that got the poo from healthy mice. So there's some probiotic for you. So our overall hypothesis for one aspect of the connection between skin and gut is, we need to think about actually, are these associations that we see between skin disease and gut disease sometimes that the skin disease is predisposing people to gastrointestinal disease? That's the first big surprising take-home message. And the mechanism by how that could happen is through this hyaluronidase and the TLR4, the antimicrobial peptides, and then a change in the gut microbial community. So that's skin-gut. And then the most important conclusion is that this goes both ways. And I'm not showing any data. It's ongoing work in the lab, and many other labs have done this too. If you do things to the gut, of course, you can also change stuff in the skin. So it definitely goes both ways. It's not a one-way street. Next is communication between skin and heart. And this was inspired by some amazing observations that were made about 15 years ago, first starting in Penn, but at another number of institutions around the world since then, showing that the risk for cardiovascular disease in patients with psoriasis was greatly increased. And actually in the one trial shown here on the left with psoriasis, talks about the risk factor for cardiovascular disease being 1.5 in psoriasis. And you will note that that's actually higher than smoking. So it really drove home to me, not only that that association exists, but this, your doctor tells you don't smoke. The doctor doesn't ever tell you today, don't have psoriasis when you're trying to work up your cardiovascular disease, but you guys should, right, with this kind of information. And these associations have been seen in other inflammatory diseases too, in particular rosacea, not as high as in psoriasis, but there's also a risk, an increased, statistically significant increase in cardiovascular events in patients with rosacea. Now that was of interest to the lab because both of these diseases, we and others had shown have too much of these antimicrobial peptides in the skin. Psoriasis long ago was shown to have tons of it in the skin, and actually others have gone and looked in serum and found that this one particular antimicrobial peptide, LL37, is about 10 times to 20 times higher in circulating serum in psoriasis patients than in healthy controls. And rosacea too, very, very high levels of LL37 in most of the patients, which we think explains the pathophysiology of the disease. And this is some overviews, and if anybody want to look it up, it's complicated stuff, but the bottom line is that the excess of antimicrobials can be bad, and the excess of this particular antimicrobial peptide can drive inflammation. I mentioned that to you earlier when we talked about the COVID experiments. And this very same guy, Gerard Wang, who had done this machine learning approach and tested why these peptides can cause increase in inflammation, had found that the LL37 antimicrobial will bind in solution to nucleic acids and make them pro-inflammatory because of a change in shape. And he does that in part by using small angle X-ray scattering approach on the cyclotron up in Berkeley. And you can see in solution how these peptides and different molecules assemble. So we asked him, Gerard, we know atherosclerosis and so forth is associated with LDL particles and lipid particles in the blood. Would you mind going up to the cyclotron and taking LDL and put it with some LL37 and see what happens? And he was all excited after staying up all night running the cyclotron, because you have to sign up for time and all of this stuff. And he came back with this graph, which meant nothing to me. It still means almost nothing except for after he explained it. But he said that peak with the arrow there in the green means that the LDL particle is getting wide and flat. It's completely changing its shape. Another peptide that is also pro-inflammatory like LL37 called LL34 did the same thing. That was sort of another duplicate positive control. But we also gave him the mouse peptide, which is called CRAMP. That's the lower one here. And the mouse peptide of the same gene family didn't do that. I'm like, that's curious. So only the human version of the gene would change the LDL particle, but not the mouse version. So is that important for cholesterol transport? We did another experiment that looked for how LDL particles get into cells. And I'm not sure that it projects, but normally LDL will bind to the LDL receptor. And when it's binding, you can see how much LDL is binding by how many sort of red dots on the surface of the cell. When you have the antimicrobial peptide there, all of a sudden these big flat LDLs stick like crazy, and they bind all over, and we show they get driven into the cell a very high amount. And then we did this experiment with many different species of this antimicrobial peptide variant. And the old world apes, like a human and gorilla and gibbon, their species version of this all did that. And actually, these are species that when they age, they do get aspects of cardiovascular disease. But many other species, like dogs and rabbits that are used common in cardiovascular disease, and mice that a lot of us use, they won't get atherosclerosis in aging. You have to do special genetic things to them. And in fact, their antimicrobial would not do this. So that made it hard for us that like to try to prove things using mice, because the mouse version of these things wouldn't be predicted to work. So flash forward another several years, we made a humanized mouse by taking the human gene for the LL37 that causes LDL shape changes and inserting it into the same position in the genome where the mouse was. So now we made a humanized mouse that looks like it makes the human version of this LL37. And then you have it go on a high-fat diet for 10 weeks. You also have to do another little genetic manipulation by deleting ApoE that makes them somewhat slightly susceptible to atherosclerosis. So we get asked a simple question: if you have too much LL37, too much of the antimicrobial, like you do in psoriasis and in rosacea, are you more likely to get cardiovascular disease? And we can do this now in 10 weeks instead of over a lifetime of an individual. And the answer to that question is yes, a lot. This is an aortic arch from the mice, and hopefully you can see all these plaques just building up in the mice with LL37, the human one, but the mouse that has its normal antimicrobial didn't get so many. And when you look in the plaques themselves, you can see with two-color staining, green is the human antimicrobial, and red is CD38. So you get this yellow expression with the LDL, that it's being driven into the plaques that are building up in the mice. And actually earlier, several years ago, some people in New York had stained human atherosclerotic plaques and found this antimicrobial there, but we didn't really think much of it because we knew it was around. We didn't think it was causal, but this experiment suggests it's causal. And in collaboration with a cardiac scientist here at UCSD, we got a number of blinded collected samples and tested, and this is not psoriasis patient, this is just a general population, is the antimicrobial peptide level at all associated with cardiovascular disease risk in the general population. And it turned out a pretty good R-square value and pretty good significance that if you happen to have too much LL37s circulating in blood, you actually have a higher risk of cardiovascular disease because it correlates with this PCOX phospholipid value. So the model there now is skin is causing problems with the heart. And if you have things in the skin that make too much of this antimicrobial, because of the way this is designed, this human antimicrobial, it can flatten your LDL particles, make them get into the cell too much, and almost act like you're a familial hypercholesterolemia patient, where you have, even though your LDL levels are normal, that normal level of LDL is driving more atherosclerotic disease. So bottom line, if you got skin disease, think about cardiovascular outcomes in your patients. And the last bit, in the last few minutes, is the most recent work looking at the communication between the brain and the skin. And there's a lot of interest in derm research now in terms of itch and pain. We've made a lot of progress in peripheral nerve communication. In skin diseases, we know neuropeptides can influence immune functions. We know that in situations of stress, so forth, immune function is different. But we felt like we didn't know everything about that. In particular, the question of infection and stress. So acne is worse than stress. Other types of infections you can get predisposed to. So Marcus Chan, who's sitting in the audience here, who will unfortunately only be with us for another month before he goes to his new lab that he's been given in China. So congratulations, Marcus. If you're a mouse, stay out of Marcus's way, because he read the literature on good models for stress in mice. And one of those models involves taking a big test tube where the end is cut off and they can breathe okay, but putting them in the test tube and locking them in that 50 milliliter tube for about 3 hours. And they don't like that. You know, it's not free wheels and running around the cage. They get really stressed out. So what Marcus did is he got protocol approval from the IRB to do this. He would go through all the work of going down there and putting some mice in these tubes for 3 hours a day for 3 days, and then looked at how susceptible they were to Staph aureus infection. And that middle picture shows you how big the lesion is when they're stressed, and it's much, much bigger. It's a significant difference. And then if you count the bacteria in the lesion, you can see that they're growing better. So these mice, when they're stressed, was a good model to show increased infection related to psychological stress. Lots of different ways that could happen, but one of the ways to test this is through the hypothalamic pituitary axis, the HPA axis, your adrenals. And you could test if the adrenals are involved by just surgically removing the adrenal. So again, stay out of Marcus's way if you're a mouse, because he might take your adrenals out, which he did. And those mice without adrenals, despite being stressed and locked in the tube, they didn't get such big infections. So the adrenal was obviously important by a surgical approach. You could also give things like, propranolol, give adrenergic blockade mix, and just the chemical mix would protect them against infection. He looked at the gene expression in the skin and saw that if you took the adrenal out, the genes that changed with stress became more normal. And then he found that if you gave one of the major stress hormones, adrenaline, if you injected that into a mouse, that mouse, all hyped up on adrenaline, became more susceptible to infection. So multiple different methods to make the point that, all right, this is the beginning of the pathway. What's the end? So the next thing he looked at is when he started examining the skin morphology during infection and the histopathology, we saw that the fat layer really changes a lot during infection and stress. And in just a normal mouse without any infection at all, stress actually shrunk the fat, which is stained here by a lipid staining dye that's in green. Consistent with what we had published back in Science a few years ago, fat is part of what helps fight infection. Remember I told you that, by producing antimicrobials. In a normal Staph aureus infection, the fat would produce these antimicrobials, and in fact, the normal Staph aureus infection showed increased green staining, increased fat. I keep grabbing my belly when I talk about it. Is that a bad sign? I think so. And then stress counteracts that, and adrenaline counteracts that. So now, we have a hypothesis we could really test. We think fat is important, at least the development of acute fat is important in infection, and stress seems to block that, and stress from the adrenal gland blocks that. So that all fits. Nice story. And this process of reactive adipogenesis, which I implied for you, says that a lot of antimicrobials are made as the fibroblast goes into the fat cell. So could the increased infection be related to the blockade of this process? And the answer is complicated and very surprising to us. And there's a lot of different data points that support that hypothesis. But I wanted to show you this since many of you are learning to be dermatologists and like to look at histopathology. This is mouse skin with a Staph aureus infection. The green staining is staining PDGF, which helps us identify the fibroblast that could become fat. And the red staining is the mouse antimicrobial, the one that doesn't cause cardiovascular disease, cramp. So in normal infection, you just get a ton of red staining. And the cramp is brought there by neutrophils and also made by the fat cells. So when it's not a neutrophil and it's made by a fat cell, you get co-staining. So it's the green fibroblast, red cramp. So you get a lot of this co-staining. So we can see all the antimicrobial being contributed to by the fat. And after stress, although there's still plenty of neutrophils around, lots of red staining, all the yellow is pretty much gone. So a major cell type producing an antimicrobial is being affected by stress. To prove that it is that cell, we have a mouse genetic approach where we could just knock out a gene in a specific cell type. We did that in fibroblasts. And when that happens, if the fibroblasts can't make the antimicrobial, they get worse infections, but stress doesn't make the infection get any worse on top of it. So it implies that it's the fibroblast effect from stress that causes the increased infection. The last part of the question is, why would this happen? Why would stress inhibit the antimicrobials coming from only fibroblasts, but leave the neutrophil ones alone? So we go back to the single cell approach, look at a lot of genes, and there was a very, very subtle signal that is shown by that red arrow, which said during stress there was increase in another factor known as TGF-beta. We went back and confirmed that, and measured the amount of TGF-beta protein in the serum, in the plasma of these mice, and saw that TGF-beta goes up with stress. That's something that wasn't known, wouldn't be expected. TGF-beta for skin is mostly been attributed to scarring. It's known to affect fibroblasts, but it really wasn't known to have anything to do with innate immunity. But if you take the fibroblasts and remove the TGF-beta receptor only on the fibroblasts, you can completely rescue the infection from stress. So saying the pathway is that stress, adrenal gland, adrenaline inhibits reactive adipogenesis because there's this release of TGF-beta, and the absence of detecting TGF-beta makes them then normal again. Very stressed out still very stressed, but they can now fight infections. So this is the model. Congratulations to Marcus and the Science Immunology paper on that, kind of going through what I just showed you. What we're kind of dreaming about now is we're learning about different immune functions of fibroblasts as a way to further reduce therapeutic side effects by targeting therapies that we think work through the fibroblast mostly to just the fibroblasts. And a good example of that is Kellen Cavagnaro's work in the lab that's shown things like IL-17 and TNF, things that we have, biologics that are really important for affecting skin disease. And Tomo, who's looking at IL-4 and IL-13, things that are really important for atopic dermatitis. The fibroblast might be the major cell type that's responsible for the disease because of the recognition of those. So we could target just those cells. And there's thoughts about how to do that. We might be able to completely change the therapeutic index of some of the drugs we use. So that's a little story, 3 little vignettes. Thank you very much for your attention. The bottom line is there's a lot of cross wires here, but if we look carefully, I think there's a potential to start sorting out these different pathways to improve diagnosis and improve disease. And with that, I will thank everybody in the lab. Thank you for your attention.